Good afternoon to all the NEET aspirants 2026. This is your Prasanna Ma'am from NEET World. So yes, today we are going to begin with plant physiology. We have completed cell biology under our 100 days concept, 100 concepts for NEET 2026. We have completed cell biology. Under that we have completed all the important concepts regarding 3 chapters. Those are cell, the unit of life, cell cycle and cell division, and biomolecules. All the important topics with respect to NEET 2026 are covered from those 3 chapters. Now let us begin with plant physiology. In that 3 chapters that we are going to look into: photosynthesis in higher plants, respiration in plants, and plant growth and development. We are going to look into this beta. So before getting into the concept, let me give you a brief introduction about NEET World's special programs. So under NEET World's special programs, we have NEET World's 2 steps to medicine for 11th standard students, and which has very minimal fee of 3333 per month, and NEET World's 1 step to medicine for 12th standard students with the same fee, and a unique program, that is NEET World's Power Step to Medicine with the same minimal fee of 3333 per month, which is feasible for all the students of different financial backgrounds. So yes, whoever are interested can make a call to this number or directly visit our branch, which is in Narayanguda, Hyderabad. This is all about our special programs, ma. Make use of it. Yes, let's begin with the concept. Under photosynthesis in higher plants, today we are going to deal with the initial things of this chapter. Those are early discoveries. What type of question we can expect from the early discoveries? Most importantly, from early discoveries we can expect to match the following type of question: asking you the scientist's name and giving you in the other column of their discoveries or the experiments that they have conducted for the photosynthesis. Second is action spectra and absorption spectra, which is delivered by T.W. Engelmann. We are going to look into it more detailed way, and we are also going to discuss about photosynthetic pigments and chloroplast as well. So these are the concepts we are going to look into from photosynthesis in higher plants. Let's begin, ma. We have few synonyms for photosynthesis. What are they? Photosynthesis is also called as physico-chemical process. It is also called as what, ma? Physico-chemical process. It is an anabolic process. And it is said to be an endergonic process. And this is an oxidoreductive process as well. Let us look into all this, like why photosynthesis has these many synonyms. You should know the reasons, because we can expect an assertion reason type of question or statement based from this. Okay, photosynthesis is said to be a physico-chemical process because the physical form of energy, that is photons, are converted to the chemical form of energy. Those are ATP and NADPH. Physico-chemical process. What does it mean, ma? Physical form of energy, that is photons, are converted to chemical energy, that is ATP and NADPH. Clear, right? Next, it is said to be an anabolic process. Why is it said to be an anabolic process? Because using the simpler substances, be it carbon dioxide, water molecules, and sunlight in the form of photons, these simpler molecules are formed or synthesized into a complex molecule, complex organic molecule, that is glucose. So it is involved in the synthesis of complex organic molecule from simple inorganic molecule. So this is said to be a what? Anabolic process. Endergonic process. In this process of photosynthesis, the energy is being utilized for the formation of primary product, that is glucose. So that is why it is said to be endergonic process, as it is utilizing the energy. And this is said to be oxidoreductive process. Oxidoreductive means what, ma? Here in this reaction, both oxidation and reduction are taking place. How? Let us look into by taking the standard equation of photosynthesis. What is the standard equation of photosynthesis, ma? 6 CO2 plus 12 H2O in the presence of sunlight and chlorophyll, in the presence of sunlight and chlorophyll. It is synthesizing the primary product of photosynthesis, that is C6H12O6 and 6 water molecules, and oxygen is evolved as a byproduct. So, from this standard equation of photosynthesis, if you have to consider where is oxidation happening and where is reduction happening. Here carbon dioxide is reduced to glucose. Can you see here beta? Carbon dioxide is reduced to glucose. So, this is the where reduction reaction is happening. Clear, right, ma? And water molecule which is in the reactant, it is being oxidized to oxygen, where oxygen is evolved as a byproduct. So, here water molecule is evolved as what? Oxygen. So, this is what beta? Oxidation. Clear, right, ma'am? So, in the single reaction here you can see both oxidation and reduction process. That is why photosynthesis is called as oxidoreductive process. Once let us quickly recall the terms of photosynthesis synonyms, like it is also called as physico-chemical process, because the physical form of energy, photons are converted to chemical form of energy, that is ATP and NADPH. Second, it is an anabolic process because it is involved in the synthesis of complex organic molecule, that is glucose, from simple inorganic molecules. Third is it is an endergonic process because in this process energy, heat energy or light energy is being utilized, and this is an oxidoreductive process because both oxidation and reduction are involved in this reaction. Now, let us look into the early experiments in detail beta. First, we will look into the early experiments, like how did they prove that carbon dioxide and water are the reactants of photosynthesis or the raw materials of photosynthesis. How did they say that sunlight is essential for photosynthesis, glucose is the primary product of photosynthesis, and oxygen is evolved from water. All these points are delivered through what? Experiments. Let us look into them one by one. Firstly we look into carbon dioxide is essential for the process of photosynthesis. Who delivered this point? This is delivered by Mole's half-leaf experiment. Mole's half-leaf experiment. So, here in Mole's half-leaf experiment, if you look into it, that a plant which is exposed to sunlight, half of the leaf is immersed into the beaker containing KOH solution. Okay, so it is immersed into a beaker containing KOH solution. This KOH will completely absorb the carbon dioxide which is present in this half leaf. So, reacting with this carbon dioxide, it forms out to be K2CO3, that is potassium carbonate. Clear, right, ma? So, half of the leaf which is immersed into the KOH solution, reacting with carbon dioxide, this forms out to be potassium carbonate, and complete carbon dioxide in this half leaf will be exhausted, and half of the leaf which is exposed to sunlight, that is containing carbon dioxide, and it normally conducts the activity of photosynthesis. When this leaf is conducted with starch test, okay, when this leaf is conducted with starch test, it shows that half of the leaf turning blue-black color, indicating the presence of starch, and half of leaf will show pale color, indicating the absence of starch. With this, it is proved that carbon dioxide is essential for the process of photosynthesis. Remember the name of the experiment, that is Mole's half-leaf experiment. Here, if you look into it, that is bell-jar experiment. Okay. Bell-jar experiment by whom? Joseph Priestley. Bell-jar experiment by Joseph Priestley. So Joseph Priestley in the year 1770 he started conducting the experiments in discovering the oxygen, but then he has discovered oxygen in the year 1774. Okay, oxygen discovery. We all know the popular bell jar experiment, in which in a bell jar a plant, mouse and a candle, burning candle is kept. After some time he has observed that 3 of them are surviving, like candle is still burning and the plant is still green and the mouse is still alive. Previously when he has conducted the same experiment with animal and candle, the candle has put off and mouse has dead. So when he conducted the same experiment with mint plant, 2 of them are alive. He said that whatever the air that plant is restoring, that is being released by mouse and burning candle. Burning candle and mouse, whichever are releasing the gas, okay, or air or gas, whichever is being released by burning candle and mouse is restored by the plant. Okay, this is what is said by Joseph Priestley, and later he named that air or gas as what? Oxygen, in the year 1774. Remember the year, scientist and the experiment name. Here you look into beta, that is Jan Ingenhousz experiment. Okay, Ingenhousz experiment. What did he convey? Jan Ingenhousz has said that water and sunlight are essential for the process of photosynthesis. Water and sunlight are essential for the process of photosynthesis, and oxygen is evolved as a byproduct of photosynthesis. These are the points which are conveyed by Jan Ingenhousz experiment, and this is a hydrilla experiment. Okay, experiment is conducted on hydrilla plant. The model organism is hydrilla. What did he do actually? He has taken a beaker containing water, and in this beaker he has taken a tistil funnel. In the tistil funnel he has kept that hydrilla plant, inverted that tistil funnel. Above the tistil funnel he has placed a test tube. Left 2 such setups, one in dark and one in light. The setup which is exposed to light has shown some water bubbles in the test tube. When this test tube is taken and tested for the oxygen, the flame has burned more vigorously, okay, indicating the presence of oxygen. So, with this he said that water and sunlight are essential for the process of photosynthesis, and oxygen is evolved as a byproduct of photosynthesis. Clear with this, right beta? Next experiment is T.W. Engelsmann experiment. This is the most prominent experiment actually, T.W. Engelmann's experiment. From Jan Ingenhousz experiment, it is very clear that water and sunlight are the raw materials of photosynthesis, and oxygen is evolved as a byproduct of photosynthesis. But from T.W. Engelmann's experiment, he clearly says that which region of sunlight is actually more efficient for photosynthesis. Which regions of sunlight? He conveyed blue region and red region of sunlight are more efficient for the process of photosynthesis, whereby with this he has delivered both action spectra and absorption spectra. Action spectra and absorption spectra. Clear, right ma? So here you look into it carefully. Firstly, how did he conduct this experiment is, a unilateral light is projected to the prism, and this prism will scatter this unilateral light into WebGR. So this WebGR light which is scattered from the prism is exposed to a filamentous green algae. That filamentous green algae is what ma? Cladophora. Clear, right? So unilateral light is exposed to the prism and then it gets scattered into WebGR, and this WebGR scattered light is exposed to cladophora. Over this cladophora, aerobic bacteria are placed. Okay, over this cladophora, aerobic bacteria are placed. After some period of time, it is observed that aerobic bacteria got accumulated in between 400 to 500 nanometers wavelength and in between 600 to 700 nanometers wavelength, indicating blue and red regions are more efficient for the process of photosynthesis. How? As the oxygenic photosynthesis is conducted by cladophora, oxygen is being evolved. Wherever more oxygen evolution is there, their aerobic bacteria gets accumulated. Okay, their aerobic bacteria gets accumulated, indicating blue and red regions are most crucial for the process of photosynthesis. With this, experimental part is done. Now let us look into the gist of whatever we have learnt over here. So look at here, Vita. Just a second. From this single standard reaction, I will give you the complete let out of all the experiments for your exam point of view. Okay, there is nothing to worry. This many experiments, how to remember, that worry not at all required. If you take the screenshot of this one slide, that is enough for early discoveries. Okay, let us look into it. So first you tell me, ma, who said that carbon dioxide is essential for the process of photosynthesis? It is by Moll's half-leaf experiment. Moll's half-leaf experiment. Right, ma? And who said that water and sunlight are essential for the process of photosynthesis? It is said by Jan Ingenhousz. Jan Ingenhousz. Fine. And he is the person who said that oxygen is evolved from the process of photosynthesis. Clear, right, ma? Who said that glucose is formed as a primary product of photosynthesis? That is said by von Sachs. Okay, it is clearly dealt by whom? Von Sachs, Julius von Sachs. He said that glucose is the primary product of photosynthesis, and this is the primary product of photosynthesis is said by him by conducting what experiment? Variegated leaf experiment. Variegated leaf is a leaf which will not have complete green areas. It will have green and non-green areas. Conducting starch test for the variegated leaf, he clearly said that only green parts of the leaf will conduct photosynthesis and help in starch formation. Clear, right, ma? So glucose is the primary product. It is said by whom? Julius von Sachs. And then oxygen is coming from water, not from carbon dioxide. That point is said by C.B. Van Niel. Okay, oxygen comes from water, not from carbon dioxide, is said by C.B. Van Niel. By what experiment, ma? He has conducted bacterial photosynthesis. Okay, he has conducted bacterial photosynthesis. Fine. C.B. Van Niel has conducted bacterial photosynthesis, saying that oxygen is coming from water. How? Because in bacterial photosynthesis, the electron donor will be H2S, and at the end products are formed like sulfur will be evolved. Same case in oxygenic photosynthesis of higher plants, where water is the electron donor, oxygen is evolved as a byproduct. Okay, so by conducting bacterial photosynthesis, C.B. Van Niel has said that oxygen, whichever is formed as a byproduct of photosynthesis, is coming from water, not from carbon dioxide. And finally, who discovered oxygen? Oxygen discovery is done by Joseph Priestley. So whoever want this, you can take a screenshot of this also, Vita. So completely carbon dioxide essential for photosynthesis, Moll's half-leaf experiment. Water and sunlight essential, said by Jan Ingenhousz, hydrilla experiment. And lastly, oxygen evolved from photosynthesis is said by Jan Ingenhousz. Oxygen is discovered by Joseph Priestley. Thirdly, oxygen is coming from water, not from carbon dioxide, said by whom? C.B. Van Niel. Remember these three important points. Don't get confused if you are getting the question on oxygen discovery, evolved from photosynthesis. Thirdly, as a byproduct coming from water. Look into each and every experiment and scientists. They are different, beta. And glucose formed as a primary product is said by Julius von Sachs. Clear, right? So with this we have dealt or completed with the early discoveries part. And next if we see, yeah, photosynthetic pigments. Before going to photosynthetic pigments, let us continue this T.W. Engelsmann experiment. Experiment is clear to us, and let us understand the action spectra and absorption spectra. First, action spectra. What is action spectra and absorption spectra? First, let us look into both of them. Absorption spectra is this, wherein in this absorption spectra it clearly delivers you that different pigments absorbing light at different wavelengths is said where? Through absorption spectra. In absorption spectra, what we can see, ma? Different pigments absorbing light at different wavelengths is said in the absorption spectra. What is action spectra? In action spectra, you can clearly see only that one pigment which conducts maximum amount of photosynthesis by absorbing maximum wavelength of light. Okay. So, different pigments absorbing light at different wavelengths, you can see it in absorption spectra. Only one pigment which is absorbing maximum amount of wavelength and conducting maximum photosynthesis is said in action spectra. That is the difference between absorption spectra and action spectra. Now, let us look into the photosynthetic pigments, and then we will go to the clear analysis of absorption and action spectra. Photosynthetic pigments, if you see, ma, most leaves will appear green because they reflect green light and they absorb blue and red regions of light. Whichever light is reflecting, we can see only that color, right? So, this is reflecting green light and it is absorbing blue and red regions of light. Some leaves are different colors, are of different colors due to the presence of different pigments. Pigments can be carotenoids, xanthophylls, phycobilins. So, these are different pigments, of which carotenoids and xanthophylls are fat soluble pigments. Okay. Whereas phycobilins are water soluble pigments. Okay. And we have chlorophyll A and chlorophyll B. As such, we have different colors, beta. Chlorophyll A is of what color? Chlorophyll A is of blue-green color. Blue-green color pigment. Chlorophyll B is of yellow-green color. Carotenoids, orange color. Carotenoids are orange in color. Xanthophylls, yellow in color. Fine, ma. So, here chlorophyll A blue-green, chlorophyll B yellow-green, carotenoids orange and xanthophylls are yellow in color. Remember the colors of the pigments. Here I would like to tell you one more point about chlorophyll pigments. Chlorophyll pigment or chlorophyll structure will be as such key. It has one porphyrin ring and a phytol tail. This porphyrin ring will be having magnesium ions. It will be like this, ma. Yes. So, this is porphyrin ring, which is a tetrapyrrole ring which is connected to magnesium ions at the center. Okay. And this has a tail. This tail is called as what? Phytol tail. Clear, right, ma'am? So, here you have chlorophyll is having what? A porphyrin ring which is a tetrapyrrole ring and it has a phytol tail. Clear, right, ma'am? So, here it has different pigments and note down the colors of that and the chlorophyll structure, chlorophyll pigment structure. Amongst the constituents of white, light, red and blue wavelengths, the best to carry out photosynthesis. Just now we have discussed that red and blue wavelengths are best to carry photosynthesis. This is inferred by what experiment? T.W. Engelsmann experiment, and the model organism used is what? Filamentous green algae, that is Cladophora. Now, let us look into the action spectra and absorption spectra. So, here you see, ma, here in absorption spectra, what pigments are tabulated? Chlorophyll A, chlorophyll B and carotenoids. This is the direct image of your NCERT. Focus and listen, beta. So, you have chlorophyll A, B and carotenoids. Chlorophyll A will show two peaks, one in between 400 nanometers to 500 nanometers and other in between 600 to 700 nanometers. And here one more point I have to say you that this pigments or the wavelength of light, usually the visible spectrum of light, visible spectrum of light will be of 390 nanometers to 720 nanometers. Of this, photosynthetically active radiation will be PAR, that is photosynthetic active radiation. PAR will be of 400 nanometers to 700 nanometers. Clear, right, ma? The visible spectrum of sunlight will be 390 nanometers to 720 nanometers. Photosynthetic active radiation, that PAR, where the maximum efficient photosynthesis is conducted, it will be from 400 nanometers to 700 nanometers. As such, here in absorption spectrum you can see chlorophyll A has two peaks, one in between 400 to 500 and one close to 700 nanometers. Chlorophyll B also shows two peaks, but they are in between 400 to 500 nanometers only. Sorry, chlorophyll B one peak and carotenoids two peaks are shown. But here if you can observe properly, the maximum amount of photosynthesis is conducted by chlorophyll A. How can you say that, ma'am? So, here you can see two highest peaks, okay, two highest peaks at two different wavelengths only by chlorophyll A. That is the reason chlorophyll A is said to be the core pigment and the rest pigments are what? Accessory or antenna pigments. Clear, right now? So, here this is about the chlorophyll, and in action spectrum you can see, okay, action spectra, that chlorophyll A is conducting maximum photosynthesis. And here in this graph you can clearly observe that chlorophyll A of the absorption spectrum is coinciding with the action spectrum. Look into this, ma. Maximum absorption of light is by chlorophyll A in blue and red regions. This shows higher rate of photosynthesis. That is why chlorophyll A is considered as what? Chlorophyll A is considered as core pigment, right, or reaction center. Here, hence we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. Okay, chlorophyll A is the chief pigment which is associated with what? Photosynthesis. This is all about action spectrum, absorption spectrum and this graph. Clear, right, ma? So, with this I will end today's session. Tomorrow we will be looking into the chloroplast structure, light harvesting complexes and photosystems, and we will also look into more keenly Z scheme, that is non-cyclic photophosphorylation, from which you will get a definite question in NEET 2026 without fail. Okay, so that we will look into tomorrow. And before ending the class, I would like to once again brief you about NEET World's special programs for 11th, 12th standard students and for long term students as well, wherein in NEET World we have 2 steps to medicine, 1 step to medicine and power step to medicine with the minimal fee of 3333 per month, wherein in this fee only you will be getting the complete study materials, complete lecture content and 2 master revision strokes for your exam. So, don't miss this opportunity. To know more about these courses, you can call us to the given number or directly visit the branch which is in Narayanguda, Hyderabad. And to all the students who are studying in the public platform for your self-study, here we have a Telegram group wherein we will be guiding you in each level of your preparation. How? If you can join this Telegram group through the QR code, by scanning QR code, you can attend daily quizzes of biology, chemistry, physics and mathematics for JEE also. And live class schedules can be updated in this Telegram group. Free resources of questionnaire PDFs might be, study materials might be are provided in this group, and one-on-one doubt solving session with the faculties are also provided. And to know more about the courses and the institute, you can call us to the number or you can directly visit our official website and log in to the Telegram group. Thank you and see you tomorrow.